This video is an introduction into Hotforest Enterprise and its current projects. Hotforest aims to innovate technologies and services that advance the circular bioeconomy, which uses bio-based materials to close production loops and eliminate systems-level waste. Hotforest was formed in fall of 2020 by Mark Rudnicki, Professor of Practice in Forest Biomaterials. There are three concurrent projects in our enterprise currently: the Solar Table Project, the Mycelium Project, and a Senior Design Project. We have weekly full team meetings on campus in addition to project work time in the SAMS pilot plant. Typically, a product has a linear life cycle where it is created, used, and then discarded into a landfill. The circular bioeconomy aims to design waste out of the system through recyclability, creating a second life for products or products made of renewable bio-based materials that can be composted. Using bio-based materials will allow the circular economy to become more attainable as decomposition becomes an option. Hotforest Enterprise is made up of one team consisting of two crews as well as a senior design project, as mentioned. The Solar Table project began in fall of 22. By the end of the fall semester, an initial prototype was completed. This semester, a second iteration of the Solar Table has been completed. The Mycelium project began in spring of 2023. The purpose of this project is to reduce wood waste in furniture manufacturing and use mycelium as an alternative. Furniture production produces high amounts of waste from cuttings and post-consumer waste, which is often discarded into landfills and contain harmful adhesives. Our team has grown mycelium to bind to wood particles to create a brick-like structure which can be used for sustainable alternatives in packaging, construction, furniture, and other materials. We use a control substrate of clean aspen in two different particle sizes, along with a substrate of ground furniture waste cuttings. Our process involved 5 main parts: preparing the substrate or growing medium, expansion of cultures, sterilization, inoculation, and the colonization or growth period. We have collaborated with Michigan Tech's Wood Protection Group and Tara Ball on this project. The Wood Protection Group helped our team by allowing us to access the mycology lab and prepare our mycelium cultures. We chose aspen as our control substrate due to the species of mushroom spores used. Here we have aspen wood ground to 2 different particle sizes. For the large size, we use our Filmmaker tabletop grinder and ground the wood pieces 8 times to produce the results you see here. A finer particle size was used to promote faster growth as well. For the finer particle size, we use aspen sawdust and then ground it to a particle size of 4 millimeters. To promote circularity, we chose to use wood furniture waste from Steelcase Office Furniture Company as a substrate. This waste does not include plastic. The black components on the furniture waste pictured is paper. This was also ground by the Filmmaker 8 times and then ground on the 4 millimeter blade to produce these results. When working with mycelium, it is crucial that all materials are sterile to avoid contamination. Before starting any work, our fume hood was sprayed with bleach and an alcohol solution and wiped down. We also made sure to spray our hands with the alcohol solution and that all tools were dipped in a sanitizing solution and were flame sterilized before use. To sterilize the substrate, each type was sterilized for a full cycle of 45 minutes in an autoclave, which is a machine that kills potential contaminants using steam pressure. To inoculate our cultures, we placed each type of our substrates into 2 grow bags per type. We added 1 petri dish of prepared mycelium into the bags containing the wood substrate. Then we added 300 milliliters of distilled water to each grow bag. The 6 grow bags containing the substrate, mycelium culture, and distilled water were then transferred to a room with a controlled humidity of 85% relative humidity and a temperature of 80 degrees Fahrenheit and left to grow. We monitored the growth by checking on the inoculates every 2 days during the growth period, documenting the rate of growth and the moisture level in the bags. The white appearing on the bags in these pictures is mycelium. The team is planning to extend our research into next semester and explore the use of mycelium in products such as furniture components, sustainable packaging, and adding structure and bonding properties to recycled wood fibers. We are working with IKEA to discuss next steps for the project and possible funding as well.
We are the Solar Table team. Included in the picture is our team. Going from left to right, we have Dan, Nicole, Josie, and Dominic. We are a multidisciplinary team, with Dan and Nicole being from the College of Forest Resources and Dominic and Josie being from the College of Engineering. As more businesses and people use solar energy, there's an increasing number of solar panels that are being sent to landfills when they no longer serve their purpose. When the panels are retired, they often still have life left in them. With new solar technologies that are being developed, such as Solar 4.0, that are more efficient, it will make existing solar technology obsolete. We are creating a way to divert these obsolete panels from landfills by extending their life and creating something useful. The first step in this process was to create a CAD model, after searching the internet for some adequate design parameters. This was done so that we were able to look at the stability of the table, where improvements may be immediately needed, and what sizes of the materials that are needed to be acquired to construct the table. After this, we had to collect wood that was to be used. To advance biocircular efforts of our table, we used reclaimed wood. The wood that we used for this project was Douglas fir for its strong mechanical properties. Using this wood also eliminates additional waste that may be created by using purchased materials. We then cut the pieces needed and indicated where necessary markings may be needed be placed on the wood for assembly. The holes were placed strategically to efficiently distribute weight across the frame. After this, the frame of the panel was then reinforced on the corners so that the weight of the panel would be evenly distributed into the frame. The frame is attached to the panel with 2 contact points on each end and 1 center support beam. The frame and the panel are one cohesive unit. The electrical box was attached to the table in a way to best suit the user of the table. Convenient, but not in the way. The solar table is designed to be simple and easily repairable, including our battery charging control systems. Inside the electrical enclosure is an MPPT solar charge controller, which handles the fluctuations in the panel output voltage to provide clean power for charging with connected batteries. The enclosure contains 2 lithium-ion battery cells for a 24-volt architecture, which feeds to the USB-C power delivery ports for users to charge their devices. The MPPT controls when to charge the batteries and regulates the load from the USB ports, allowing the table to charge connected devices and its internal batteries efficiently. When increased demand is put on the USB ports, the MPPT is configured to automatically balance the load between the solar production and batteries reserves. If either the battery or the panel stops producing enough energy for the demand, power output is automatically disconnected, preserve, and charge the batteries. With the table completed, it can be placed just about anywhere outside so that it can be used. We would like to display them around campus for students and faculty to be used. We are currently working on making a 3rd and 4th generation of the solar table with a forward focus in mind. We hope to improve the design when it comes to the materials used, the design of the frame and electronics used, and the ability to transport the table. We hope to see that these tables are used around our campus as well as others once we are further along in the manufacturing process. We also hope to find a sponsor to advance our efforts.